No, but it's gonna be hard, buddy. Race cars,
Huh?
Actually, I think I think oh yeah. There was a thing actually no, you know actually. Bro, I have to take a photo of that.
Are we all set? All right. Sigrid. Do we have all the virtual students accepted? All right. All right. So I think we're ready to get going here. I am very happy and pleased to introduce Professor Ritu Raman here from the Mechanical Engineering Department at MIT. She's won numerous awards. I'm not going to spoil her talk because there it is on the title slide here. It is just really—
Recording in progress.
And think about the innovation that's coming from looking around you, observing what you see in nature, and then putting it to work and building something similar to that, or innovating on that. So we talked about innovation a little bit yesterday. This is just another way to innovate and come up with new solutions to problems. So thank you very much. Let's hear it for Professor Raman.
All right. Hello, everybody. Super nice to meet you all today. I will tell you all about robots. I'll maybe start a little bit by just talking about my path to MIT. And one just sort of clarifying note about questions. I would say, like, as we're going through the talk, if there's something that you're just like, I'm probably confused about this, maybe other people are, it's a clarifying question, feel free to raise your hand, interrupt, and like, let's make sure we get it straight right there, rather than waiting 40 minutes till the end of the talk. And then if you have something that you think is like a deeper, longer discussion, maybe you could save it for the end. So maybe let's use that policy for question and answer if that works. But yes, hi, my name is Ritu. I am an assistant professor here, which means I'm like the babyest rank of professor. I just started a couple years ago. Before that, I did my high school in Hamden, Connecticut. After that, I went to undergrad at Cornell University, where I majored in mechanical engineering. When I was an undergrad at Cornell, I had a couple experiences that got me really excited about research, particularly research in the interface of biology and engineering. One was the first summer internship that I did after my freshman year. So it was my first official job, where I got a job volunteering in a biology lab. That was the only thing that I could find for that summer. And it wasn't really something that was interesting to me. I was mostly like a physics, mechanics kind of person. But I took this job, and the biology lab was basically studying how rats responded to different kinds of exercise training and dietary changes in their daily life. So specifically, the question they were trying to understand is, people who drink a lot of alcohol or like abusing alcohol have severe degeneration of their skeletal muscles. They have a lot of atrophy and they're very weak. And we also know that exercise in general is like good for our muscles and can make us stronger. So they were essentially trying to ask, like, could exercise be used as a therapy to help people who have lost muscle because they've abused alcohol? And so my job was to give rats a lot of alcohol and then put them on treadmills every day, multiple hours a day for a whole summer, which is just like a completely ridiculous way to spend your summer. I was like in a basement with these little rats, and they're like, oh my gosh, I can't walk. And they have tiny little treadmills, and you got to put them on there and watch them and weigh them and see how they're responding to the training. So the first part of it, I think I was kind of depressed because I was like, this is pretty lame, not a great job. But then over the summer, I started seeing the rats' bodies really adapt to different types of diet, different types of exercise, and understanding a lot about what was going on inside the system, right? And at the same time, I was thinking about the robotics classes that I was taking at Cornell, and thinking about the fact that whenever, you're sort of building with traditional materials, right? Like metals, ceramics, polymers, things that are pretty much everything in this building around us, every time you have to, a new challenge to face, like you have a bigger hill for your robot to climb, or it falls and it breaks, you have to rebuild it, right? Like buy a new part, do all of these other things. And yet these animals, right, because they're made out of biological materials, were able to adapt to changing situations without any necessary like external interventions. I didn't have to make a new leg for the rat for it to change how it was adapting to its situation. So that experience, I think, really got me super excited about biological materials and how they respond to their surroundings. And I decided that I wanted to do more research in that, and I got really excited about the idea of research. So I did my PhD at the University of Illinois in the field of using muscle as an actuator. So an actuator is something that converts some form of energy into mechanical energy or force, right? So using muscle as an actuator for soft robots. And then I did a postdoctoral fellowship, which is kind of like the PhD equivalent of a residency for med school here at MIT, where I focused on developing a lot of implantable devices that you could put inside animals and humans eventually to restore their health after trauma. And now my lab here at MIT, we started about a couple years ago, kind of combines both those aspects of robots and medicine, and particularly looking at how engineers can start building machines with biological materials. So that's kind of the overall story of what I'm going to tell you today. But I figured it'd be helpful to start with the path of how I got there first. Okay, so now the big picture of what we're going to talk about for the most part today are actuators, biological actuators. So pretty much across the world, right? Of course, all of us, but also in the water, on the land, in the air, everything that we see, and every animal, fish, bird that moves pretty much uses the same motor control system to accomplish this goal. So you have at the base muscle, which is generating force and producing motion. You have neurons in your brain and your spinal cord, so upper motor neurons and lower motor neurons that are controlling when your muscle turns on or off. And then you also have built-in sensory neurons in your muscle and upstream that are sensing like how much are you stressing, where are you in space and time. So if you have these three components, you can basically decide that you want to reach for this, pick it up, throw it at somebody, catch it, all those sort of things are because of those three main things. You need actuation, control, and sensing. And we've done a lot of things as engineers to use synthetic materials like metals and polymers to build those things, right? We have robots that move, we have machines that move, and they're pretty good. But unfortunately, to date, they're not as good as biological materials. They're not as sustainable, they're not as efficient, and they can't adapt to their surroundings, right? So if I pick up my laptop and I throw it on the ground, I guess I'll just go buy a new laptop, right? That's my solution. But if I fall and hurt myself, as you can see clearly has happened recently, eventually I can get back my original function, right? And that's what makes biological materials interesting, and that's what we want to do. So our solution, our lab solution to this problem is to think about how we can build these sort of biological actuator systems from scratch, using cells as building blocks, as Legos, build these sort of systems from scratch and use them for applications in medicine when people are recovering from trauma or injury, or use them as something that could be a functional component of a robot. So my lab is kind of broadly focused on these themes. We make robots for biology was one theme, which we basically make little mini robots to understand a little bit more about biological systems. We use biological materials to make robots, which I'm lumping under biology for robotics. And then usually when we want to be able to build any new thing, we need manufacturing equipment, right? So we also build a range of different types of 3D printers that can be used to pattern living cells. I can't talk about all of these things today, so I'll try to touch on some of those themes, but if you have questions about all of the work going on in the lab, I can certainly answer them. So we'll start with biology for robots. Okay, so very complicated graph here, but I'll give you some high-level takeaways. So what this graph is doing is talking about how much force an actuator can produce as a function of its size. So on that top right corner are a lot of things that you may have seen in your various camps and other things. Like you might have seen pneumatic actuators, like sort of soft, flexible things. You pump them full of like gas or liquid and they inflate, and then you can get some sort of actuation. There's a variety of different types of actuators. So most things kind of function at the centimeter scale, things that we can hold and breathe. Muscle is really nice because single cells are usually on the order of microns in size, right? So a millionth of a meter. So they're much smaller, but you can stack them together to make something bigger. So our muscles are centimeter to meter scale, I guess, depending on how big of an animal we are. But each cell, each individual actuator is micron size. So it's very scalable. So that's very nice. It's very efficient and sustainable because the power source for muscle is not a battery or gasoline or anything like that. It's literally just sugar. And also, this chart doesn't account for the size of the battery. So sugar is a very dense, sustainable energy source. So that means that when you look at how heavy something is compared to the battery required to sustain it, muscle is super, super efficient versus if you look at a pneumatic actuator, you know, it might look like it's really small and it's pumping around and doing things, but there's usually a big gas tank off to the side that you're not accounting for, right? And so that makes it a little bit harder to use. So this is why people started getting really excited about the idea of using living muscle as an actuator for robots. And when I started my PhD, there were some very, very basic examples of little robots that a lab at Harvard run by Kip Parker had made. So around 2007, I guess I was probably still in high school at this time, the very first kind of idea of this came into play where Adam Feinberg had this, what you're seeing there, that sort of yellowish looking thing is a thin piece of polymer. So it's basically like cling film. On top of that cling film, he's growing cardiac muscle cells, so muscle cells from your heart that sort of like beat in synchrony. And so every time those cardiac muscle cells beat, you see that film kind of start to move a little bit. So that was the very first demonstration. Then what Kip Parker's lab did, I think this paper was by somebody named Jenna Naroth. She was like, okay, that's a cool idea, but why don't we take inspiration from shapes and forms of animals that are available in nature? So she decided to use that same concept, but make it kind of look like a jellyfish. And by making it look like a jellyfish, it also started swimming like a jellyfish. And she was able to sort of increase the efficiency, the swimming speed of those machines. And then most recently, the most recent paper, they made literally a fish that swims as fast as a normal fish. And the way that works is that on one side of the fish, there's one muscle, an agonist muscle. On the other side, there's an antagonist muscle. So that's kind of like your biceps and triceps, right? And so when one contracts, then the other one contracts to help this one relax. And it was just going fast. And they were able to make it swim and move around. So this kind of stuff got me really excited about biology for robots. But you know, cardiac muscle is nice. It's very easy to work with. It generates very large forces, which is why people kind of started working with it in the first place. But it's not what nature uses for actuation. And this is for a few different reasons. So cardiac muscle is for your heart, right? Your heart is designed to always work, always be pumping. So you can kind of make it pump faster or pump slower, but you can't turn it off. So you don't usually want a robot that you can't turn off. The other thing is that your heart muscle cells, they're not designed to recover from injury, right? This is why heart attacks are a real problem, because we can't really regenerate from there and they're not in your periphery. So that's also not great for a robot, because usually robots are out in the world and they're doing stuff and they might get nicks and tears, and you want them to be able to heal after injury. And then the final thing is that, you know, skeletal muscle is really designed for neural control, precision control. So your heart, when one cell beats, they all beat. That's how it's designed to work. They're all electrically connected. But if I had that happen for actuation, that would not be great, right? Like imagine I wanted to lift this, but if I had to do that, I also had to jump every time. Like that would be really silly. So you want to be able to very precisely control which muscle you're controlling at which time. So skeletal muscle kind of can do all of these things. And maybe you've covered this a little bit in your biology classes. I honestly don't remember what I learned in high school biology, so I'm going to tell you anyway. But each skeletal muscle cell is a very, very long cell. So we call it a fiber, and has multiple nuclei. So we call it multinucleated. So these multinucleated fibers have two proteins in them, actin and myosin, and that's what you're seeing in that corner there. And actin and myosin slide together when they receive an impulse from an upstream nerve, usually like an electrical signal. So they slide together, and when they do that, the whole fiber shortens. And if you have many of these fibers stacked together, you can get a large motion. And each of these fibers is controlled by an upstream motor neuron that's sitting in your spinal cord. So one neuron usually controls, it could be like 10 muscle fibers if you need really fine motor control, like in your hands. In your legs, it could be like thousands of muscle fibers are being controlled by one motor neuron. So that one motor neuron and the fibers it controls are called a motor unit, and then many motor units work together to form a muscle. So the question is, how do we build something like this from scratch in the lab? And turns out it's really hard, because it's taken me 10 years and I'm not done yet. But I can tell you where we are and where we're going with this. Okay, so first, we need to be able to make the muscle, right? And we've gotten pretty good at that. So I'm just giving you a sense of scale here. So that scale bar is 1 millimeter. So that little robot thing that we're making is 6 millimeters, so about this big, right? Between my little fingers here. So pretty tiny, but usually something that you can see with your visible eye and very easily capture with a microscope or even a normal camera. So the way we make it is we 3D print a skeleton for it. And the skeleton, though it's kind of hard to see in this top-down view, is basically like two posts like this, and then there's like a beam connecting them. And then around those two posts, we wrap skeletal muscle tissue. The way we make that skeletal muscle tissue is essentially like making Jell-O. You mix together cells in a liquid that sort of starts off liquid and can turn solid in response to a temperature change. So literally just like Jell-O, has very similar mechanical properties to Jell-O. So very hydrated, very soft. So cells can sort of sit there and remodel it, and it sort of feels like their natural environment. And that Jell-O contains a lot of proteins that mimic their native extracellular matrix. So the native matrix that they're sitting in inside the body. So when we do that, the cells, which are shown in blue here, that's the nuclei of the cells. Initially, they all start off as single cells, and then they proliferate, right? So they divide and they grow, and they divide and they grow, and they populate this whole matrix. And then when they get close enough to each other, these are muscle cells. We start off with muscle stem cells. So the cells then fuse together to form those multinucleated fibers that I talked about in the previous slide. So those green lines that you see, each of those is a single long muscle fiber. So each of those is a single actuator. And you can tell that they're all kind of pointing in the same direction. And that's very interesting because that's what you want, right? That's what you want in muscle. Usually in your muscles, you have all of your fibers pointing in the same direction. So when they contract, you're pulling in this direction. If they were all pointing in different directions, even if they were all contracting, you wouldn't get any net outcome, right? So you need all the forces to be generated in the same axis. But we didn't plan for this to happen. And it turns out that the reason this happened is because the skeleton we made is like a little bit stiff, right? It's like a spring. So it's kind of stretching that tissue. And because it's stretching the tissue, it's imposing forces on the cells. And most cells in our body are responsive to forces. And so they align along that axis of tension. And then all the fibers are pointing in the same direction. Okay, so just, it looks like muscle, which is good. This is exactly what we want to see. But we also want it to act like muscle. And I mentioned before that usually muscle contracts when a neuron fires, right? The neuron that's controlling it. We don't have neurons in this yet. The other way to do it is to just mimic what a neuron is doing. So what are neurons doing? They're depolarizing or firing, and all that means is they're just electrically stimulating muscle cells. So one thing you can do is put muscle tissues between two electrodes and send a pulse through it, and then they will contract. That can be fine, but it's a little bit clunky to have electrodes in place, a little bit hard to like focus stimulation on one region of tissue versus another region of tissue. So what I did several years ago was work with another lab at MIT to genetically engineer our cells so that instead of a voltage-gated ion channel in the membrane, which is what's normally there, we have a light-gated ion channel in the cell membrane. So all this means is that when I shine light on it, it thinks it's getting an electrical stimulus, and then the muscle contracts. So now by shining light, and it's blue light, it's a black and white video, but it's blue light, we can get the muscle, which is that dark, dense rubber band-like thing in the middle, to contract, and then the skeleton also moves. So from these sort of things, we can understand a lot about the muscle contraction dynamics and say, okay, is this starting to look and work like the kind of muscle that I'm used to seeing in vivo, or like in our bodies and bodies of other animals? So what we see in our muscles when we track their movement is that they take a little bit of time to respond to the light. That's pretty normal. Then they contract to their peak force. Then they stay at that peak force for some period of time, and then they relax. And the really nice thing about our robots is because they're not being controlled by neurons, when they fire, they'll fire for like 5 milliseconds and then they'll stop, right? So the muscle will contract for 5 milliseconds and then stop, and then you'd have to like keep firing in order to kind of keep it in place. But with our system, because it's essentially controlled by electronics, you can keep that light on for as long as you need it to. So if you keep that light on for a longer, what we call duty cycle, so you just, the pulse width is longer. It could be 10 milliseconds, 200 milliseconds, 800 milliseconds. The muscle will contract as normal and then stay at its peak force for as long as you need it to, and then relax. So that's cool. So it acts like muscle, and we can also do things with it that we can't do with normal muscle. And the other thing we want to check is how it responds at different frequencies. So with normal muscle, when you stimulate at 1 hertz, right, 1 cycle per second, you'll contract, and then you'll have time to fully relax before you contract again. At higher and higher frequency stimulation, you contract and you're trying to relax, but before you can get all the way back down, you get another stimulus to contract. So then your dynamic range of motion is decreased, but you're still kind of moving. And then as you increase frequency, 10 hertz, 20 hertz, you're basically stimulating the muscle so much, you reach a state called tetanic contraction. So it's just like holding your muscle. So interestingly, our muscles also do that, and that's great. So that means that we can basically treat this muscle like it has all of the capabilities of natural muscle, plus some other things that natural muscle doesn't have. Okay, so similar to what I showed you in terms of swimming robots over the years, we've built a lot of different robots to try to do interesting things. So first we focused on walking. So my first paper out of graduate school, we made this sort of 2-legged robot where it's a little hard to tell, but the right leg of the robot is a little bit longer than the left leg. And so what that allows the robot to do is that every time the muscle contracts, this leg basically moves a little bit more. And so it's sort of slowly inching forward to the right, sort of like an inchworm-like motion. So that was the first robot that we built using these sort of actuators. And then the second robot we built a couple years later was making a 2-legged robot. And what this allowed us to do is that if you stimulate one leg versus another leg, you can get the robot to like walk in one direction or another, or move and turn and rotate. And then most recently, what folks in the lab have done is have the LED rather than, so all these other pictures, it's like me holding the LED and kind of following the little robot. But here, the robot's actually carrying the LED. It's like a wireless electronic. So it stimulates it, and then we can use it to basically have a completely untethered wireless machine that's like walking around and moving stuff. So that's what I've done. But the really cool thing about science is that when you discover something new and you publish it, other people can try, using your ideas and your protocols to build stuff too. So here are some other examples of folks that have tried to make little robots that swim using skeletal muscle, that pump. The pumping video is a little bit harder to see, but I think probably the easiest one to see is the gripping one. So you can see there's basically you can think of it as 2 arms, 4 muscles, biceps and triceps on each arm, and they're basically trying to pick up and manipulate an object based on muscle contraction. So these are sort of robots that people have been building. They're still pretty rudimentary because obviously the field is fairly new since, if you start it and it's only been a few years and there's sort of limited examples. But the really cool thing is that all of these videos I'm showing you are not just from the US. The Morimoto et al., for example, is from my friend Shoji Takeuchi's lab in Japan. So people all over the world have been able to replicate these protocols and build newer, cooler robots than what we were even able to do in our first round, which is great. Okay, so we can kind of build these robots, and that's nice and fun, but you want to prove that when you make a robot in this really complicated way, there was actually a point to doing that. One point could be that it's just cool and it's interesting and you want to study it. I think that's a great way to do science. Another thing also is to think about what capabilities does your material or your robot have that other robots don't have. And I started this talk by saying biological materials are cool because they can do things like adapt to their environment and sense what's going on. So the same thing is true of our robots, and we've seen that over time. So one of the first things we showed is that we could tune the strength of our actuators by exercising them. So if you shine light on the muscle every day, different periods of time, different frequencies, different durations, different intensities, a lot of optimization to be done here. But essentially, high-level takeaway, you shine light on them every day, they contract a little bit every day, they get stronger as compared to robots that aren't exercised. So that's cool. So we know that they can do that. The other thing you can regulate is their endurance, or how quickly they get tired over time. So, recently we kind of discovered that suppose we exercise our muscles at 1 hertz, so 1 stimulation per second every day for 2 weeks. And then after 2 weeks, we decide to test their fatigue, their endurance at 1 hertz stimulation. Turns out it's pretty good. You can keep stimulating them for 30 minutes, 60 minutes. They're producing the same force. They're good to go. As long as they have sugar, they're fine. However, if you stimulate them at a higher frequency, like 4 hertz, then they get tuckered out really quickly. The amount of force they're able to produce drops dramatically. And so that was kind of interesting. And some recent work in the lab that we're doing now is showing that if you train the muscle in different ways, if you train them at high frequency, then it turns out that they're actually fatigue resistant at high frequency. And why this happens, we think, is because in our body, we have a mix of slow, mid, and fast twitch fibers. So what this means is that the muscle fibers that are in your abs and your postural muscles, these are mostly slow twitch. So what this means is they produce very low forces. You don't need a lot of forces to be able to kind of hold your posture or sit or stand, but those can't get tired that quickly, right? Because you need to spend most of your day sitting or standing and kind of holding your posture. But then you have your sort of large force generating fibers, your fast twitch fibers in your arms and your legs. So that's for when I'm walking, moving around, running things. So we do need those to produce much larger forces, but they do tend to get tuckered out a little bit more quickly. And within that, there's a broad range, right? So probably many of you know, if you play different types of sports, if you did a lot of yoga versus a lot of running versus a lot of powerlifting, your body is going to look very different. And that's because those muscle fiber types are very different in shape and size and how they behave. So even though you're exercising is not one thing. We can regulate not only how strong muscles are, but also how fatigue resistant they are. And the last thing we've shown is that muscles can heal after injury. So we've gone into our muscle actuators and we've cut them, which is very hard to do because they take a lot of time and effort and money to make. And then you've taken care of these every day in a little petri dish for 3 weeks, and then you have to destroy them, which is very rough. But if you do it, what you see is that over time, these new muscle cells, muscle stem cells that are shown in green in that image there, will start to divide and proliferate across the site of the tear. and they'll fuse to form new muscle cells and fuse with the old muscle cells. And then within a couple days, the robot is basically able to completely recover from injury. So these are the kinds of things that we've done to prove to people that it is actually interesting and exciting to build robots with biological materials, because obviously now we can see that our robots can do things that other robots can't do, right? Because they're able to exercise and tune their response and heal from damage. So there are a few different things we're working on in this space right now, which I'll sort of maybe skate over a little bit. One of the things is kind of designing better skeletons for our robots. So we kind of look at our muscle as a base state and we say, okay, what is the maximum amount it can move? And then once we know that anytime you're not getting that amount of movement transferred to the skeleton that you're attaching it to, you're not actually doing a very great job of efficiently transmitting force from your muscle to your bones. So the question is basically, how do we design better bones for our robots? And I won't go maybe too much into the details of this, but the kind of big high-level thing that we're doing right now is starting to make really clever skeletons that essentially work like springs. So if you've, in high school physics, when you learn like force is equal to your spring constant times the change in length, that's essentially the same principle we're using to design really, really flexible springs from rigid materials to make very efficient bones for our muscles. So then when we attach the muscle to the skeleton, we can get very large scale movements. The other thing we're doing is thinking about how muscles are actually attached to bones. So in all of the examples I've shown you so far, we basically made a muscle tissue and we like wrapped it around something, and we kind of wrapped it around really tightly and hoped that that was enough to kind of transmit the force from the muscle to the skeleton. But that's not how it works in our bodies or any other animals. Usually there's a covalent linker between the muscle and the bone. That's your tendons, right? So if your tendons are not there, then you're going to get slippage between the muscle and the bone, and you're going to lose a lot of your efficiency. So what we've developed are these like really sticky jello-like tapes that we call tendons, but they're not real tendons with cells. They're just literally gels. But the gels are really sticky, so we can stick them to one end of the muscle and one end of the skeleton, and they're a little hard to see because they're transparent, but now we can essentially take a tiny piece of muscle, stick a tendon at either end, and then attach it to a skeleton, and then generate the same forces that we were able to generate with maybe like 4 times as much tissue. So we've dramatically increased the efficiency of our actuators and the power to weight ratio, so how much you can get done. So this is the sort of thing we're working on to make our robots better. And then the final thing we're working on with our robots is thinking about how to deploy them in 2D configurations. So I won't go into the details of this too much, but essentially what we're trying to do is think about, there's many contexts, right? Like the eye being an example, where you have sort of a 2D arrangement of muscle rather than 3D. So your biceps, for example, have very different architecture than your iris muscles. So in your iris muscle, you have a central section that's sort of circumferentially or concentrically aligned around the pupil, and then you have these radial region around the pupil. And so all of those red lines you see there are individual muscle fibers. So that's an iris that we made using our cells. And then what happens is if we shine light on the iris, we can get the pupil to open or close. So these are the kinds of things we're doing in the biology for robots space. And maybe in the remaining time I have, I'll quickly cover some of the things we're doing in the robots for biology space, which I hope will be particularly interesting if you're more interested maybe on the medical side of things than robots. Both are cool. Okay, so one quick note, I'll be showing a video in a couple slides of a mouse leg. I don't think it's particularly gross, but I'm just warning you ahead of time in case it's something that you don't want to see. Okay, so one thing that we're trying to study in my lab is not only how to make muscle for robots, but how to make muscle as an implant, right? Because as I mentioned, muscle is pretty good at healing from small injuries. So you work out too hard or you fall and you sort of rip it a little bit. It's not very good at recovering from very large injuries, right? So we know if people have been in a really traumatic car accident, they have blast wounds, that sort of thing, you lose a huge chunk of muscle. Our stem cells are not able to accommodate for that. And so one kind of idea that we and others have had is, if we're able to make muscle in the lab in a large enough size, could you implant it at the site of an injury and then have it integrate with the surrounding body so that the person's muscle can be regenerated over time? So this whole field is called regenerative medicine. People have thought about it for muscle as well as a lot of other tissues. It's a very exciting space to be working in, but I'll focus just on the muscle stuff today. Okay, so we make our muscles just as normal. and we implant them in mice that have a traumatic injury in their legs. So in the right leg of this mouse, you have a traumatic injury, you implant the muscle. And our question is, what happens if you exercise the tissues after they've been implanted? And the reason we want to do this is that people in the past have thought, okay, exercise is like generally good. So if you implant a graft in an animal and then put it on a treadmill, kind of similar throwback to what I was doing back in the day, maybe they'll get better. And this never has worked, and people couldn't quite figure out why. And I was like, well, if you think about it, when you're exercising, right, you need neurons to actually be in your muscle to be able to control that muscle. If the muscle is a graft and it's new, there's no nerves there yet. So you can't actually control it. So when you're exercising, you're not actually using that muscle. The graft atrophies, it falls apart, you overuse your injured tissue, and generally the mice get worse. So our thought was, if we implant the muscle and then shine light through the skin onto the graft, can we kind of address this problem? So the first thing I'll show you is just how our muscle fibers look compared to normal muscle. The only pictures I want you to focus on are the green ones kind of in the middle there. That bottom green one, which shows those green circles, that's the left limb of the mouse. So that's the healthy limb. That's what its muscle fibers look like in cross section. So each circle is a muscle cell. And then if you looked at it in the long axis, it would be a fiber like this. So that's what they look like, really dense, tightly packed tissues. And then the panel directly above it, that image is our graft. So high level takeaway is that our muscle and the mouse muscle look very similar, right? Okay, and then the only interesting kind of different thing about our muscle is that our cells are genetically engineered, so they have this ability to respond to light. So what this means is that by shining light through the mouse's skin, I can non-invasively control how that muscle is contracting. And so it looks like the mouse is like doing a little kick here, but the mouse is not kicking in the way that I'm kicking, because I'm thinking about kicking and I'm doing it. The mouse is not thinking about kicking. We are controlling actuation through light stimulation. And so what we see is that when we do this every day, the muscles are as strong as native tissue within about a couple days. And then we also test how long it takes the mouse to run from like one end of a room to another end of a room, and we time them. And we look at it in ones that have been injured and then ones that have been treated with our grafts. And what we see is that the ones that are treated with our grafts can basically run at pre-injury speeds within about 2 weeks. So in 2 weeks, they're completely healed, right? Which is really, really fast actually for an injury. And it would be very different if it was like a human and a much bigger tissue and there's other problems to sort out, which I can talk about later. But high level, we've understood that if you exercise grafts after implanting them, it appears to improve the rate of recovery after injury. And we did a lot of biological characterization to understand, why this might be. And what we found out is that if you look at the tissues that have been exercised and look at the tissues that have not been exercised, the tissues that have been exercised seem to upregulate signaling related to the growth of new nerves and new blood vessels into the graft. So what this means is that not only is the muscle stronger, but the host, right, the mouse is able to grow new blood vessels and new nerves into the graft potentially faster. And so that could be the reason that the mice seem to be recovering more quickly. But whenever you do an experiment in an animal, on the one hand, it's very useful because that's the main thing you have to do before trying something in a human, just to prove that it's safe and maybe worth it. However, it's also very hard to prove something in an animal because there's a billion other cell types in there, right? So I could tell you that, the reason this mouse got better is because the muscle contraction helped nerves grow into the graft, and that's why the mouse was able to use the muscle. But in order to prove it, you can't just do it in an animal. You usually also have to do it in a petri dish environment that's a little more controlled. You know exactly what's going on. So that's the kind of thing that we're working on right now, which is to think about whether we can grow muscles that are shown in red there and neurons that are shown in green, grow them in the same petri dish and watch how nerves grow out to form functional connections with the muscle fibers, and then look at how exercise sort of impacts the rate of nerve growth. So I'm not going to dive maybe too deep into this. I can talk about it a little bit more later if we want. But the high level I want you guys to look at is the top row is taking daily pictures over 5 days of neurons that are shown in this green ball here. So in the green ball, there's a bunch of neurons, like hundreds of neurons. And it's just their cell bodies, their nuclei. And then over time, like if you've seen neurons, they sort of grow out and they form these long connections and they attach to different tissues. So you can start seeing them sort of start to put out different projections over time. But compare that with neurons, same type of neurons that are grown in the presence of muscle exercise. So muscle is secreting a lot of factors like different proteins and RNA that's making those nerves really happy. It's moving and stretching them around, and that's making them really happy. And so you can see that they grow just a ton more, a ton faster and a ton farther than they would in the absence of exercise. So these are the sort of experiments that we're doing to sort of prove to people that it is really important to think about how exercise is not only good for muscle, but also communicates with a lot of other tissues inside of our bodies. And this is something that's of sort of increasing interest, just in general, right? Like we've seen sort of anecdotal things of exercise is good for your body overall. And that is true, but we're still in the process of understanding like what is the real mechanism? Like how is exercise making your immune system better? How is exercise helping your nervous system be better? How might exercise help your mental health? Like these might be very different things. So these are the sort of experiments that we do to try to understand not only how to help people recover from trauma and injury, but also how we can sort of think about leveraging exercise as a therapeutic concept. So maybe the last thing I'll show you, I'm trying to think how to wrap up. Okay, last thing I'll show you is maybe just this video that I took on a flight recently. And I want to use this to kind of motivate why neural control is important also for robotics. So that gray thing that I'm wearing there is a wristband that I wear to control nausea. There's been a lot of clinical studies that show that if you stimulate this nerve and you get really nauseous or motion sickness like I do, that seems to control some pathway that prevents you from throwing up. It works in chemo, works for a lot of people. So I gave it a try. And turns out it works pretty well for me too. But sometimes if you turn up the electrical stimulation like a little too high or you place it a little bit weird, you hit a motor nerve instead. And so that's what you're seeing there. That's my thumb moving, not because I'm trying to move it, but because the wristband is basically stimulating the muscle to contract. And I'm showing you this video because that didn't hurt, right? It was actually like, you would think that to stimulate electrically, like you'd have to stick an electrode in somebody and like it would really hurt and all those things. And that would be true if you were trying to activate the muscle directly. But the reason we like neural control is that it's really low power and really efficient. So through my skin in a wearable, I'm able to activate muscle using this very low power technique. And so this could also be useful for robots, right? Because if we can integrate neural control into the muscle actuators we've been building to make robots, we can now make robots that are incredibly power efficient. They need, even less sugar, even less batteries to control the electrical or optical stimulation. And then we can make robots that do more fun and cool and interesting things kind of out in the real world and not just in our labs. So with that, I'm gonna maybe I'll wrap up maybe by sharing a video that I really like from Boston Dynamics before I take questions. And let's see if this plays. It doesn't always play for me. Okay, let me see if I can get you to do things. Okay, you know what? Maybe I'll send you this link, the organizer, so that you can see it later. But it's a really fun video. It's like a couple minutes. It's set to some really poppy techno music. And the reason I'd like for you to watch it is that it sort of tracks what Boston Dynamics robots have looked over the past like 40 or 50 years. So I'm assuming you guys have seen a lot of the like robot dog videos and some of their humanoid videos, and they're like really cool, right? They can do backflips and they can do all these other things. And you might be like, Ritu, your robots are cool, but they're like tiny and in a petri dish and like they're not that exciting compared to these. And I would say you are right. But I would say we are with biological robotics kind of at the very start of a field, right? So in this video, you kind of see where Boston Dynamics was 40 or 50 years ago. And they have this little thing on a pogo stick that's sort of like hop, hop. And then over time, they sort of keep building complexity, more and more complexity to these robots until they get to the really awesome videos we see here. So what we're trying to do in my lab is start off where Boston Dynamics was 40, 50 years ago. And we hope we can build even more complex and exciting robots than we have today. So with that, I'll wrap up by reminding you all that science is a team sport. So these are some of the people, not all of the people. Our lab is rapidly growing from a variety of different backgrounds in mechanical engineering, material science, electrical engineering, biology, chemistry. We have a lot of different people from different backgrounds who work in our lab. and make all of the cool science that I shared with you today. And we're also funded mostly by the government and by tax dollars. So a great reason to motivate kind of thinking about where science comes from, who does it, and who it impacts. All right, happy to take any questions you have.
Thank you. Because of time, we're going to do 3 virtual questions, and then we're going to move to in-person. The first virtual question, referring to the biology of robots section, is how are cardiac muscles on the film kept alive?
Yes, oh my gosh, sorry. I should have said that for all of these things. So yes, all of these cells are alive, right? So you can't just keep them like in air in a petri dish. So cells usually need to be warm. They need to be in a sterile environment, like devoid of like bacteria. They need sugar, they need amino acids, they need a lot of different things. So every video that I've shown you for the cardiac muscle and the skeletal muscle, they're always sitting in a petri dish filled with liquid. The liquid is warm, it's sterile, it has sugar, and it has some essential amino acids. So they're always in a liquid environment. If we're thinking about, more broadly, if we wanted to deploy this in an untethered robot, something that we could live and use and work with, then you would basically need to start thinking about all of the other components that keep our cells happy and alive, right? So you basically need to build a skin that can protect it from contamination, keep it warm, and do all of these things.
Thank you. Are there any benefits to adding nerve to these robots? If so, is it ethical to make robots feel pain?
Yeah, that's a great question. So I would say, nerves, there's many kinds of nerves, right? So motor and sensory nerves are usually things that are in your periphery, which means that motor neurons are basically all they're doing is turning on or off. And sensory neurons, they are sensing stretch, right? So they can kind of figure out if a muscle is contracting. And that's useful when you need a bicep to contract, and then the tricep knows the bicep is contracted, so it can help you relax again. So those sort of motor arcs require nerves, and that's why we're starting to think about using them in our robots. However, if you start thinking about going farther upstream to the neurons in your brain, things that are capable of consciousness or feeling pain or understanding pain in some way, that I think there's maybe less justification for incorporating in a robot. So that's something that we think about a lot. We've published and talked about and worked with a lot of bioethicists in our work. So we usually think about using cells that are more focused on a specific on or off type function rather than like consciousness or pain.
Thank you. And our last question is, do you have any internship opportunities for high school students?
Yeah, so we usually, I would say for high school students, we usually have at least one high school student in the lab at any given time who has not yet graduated. So when Karina graduates, she's up there somewhere, we will have an opening, which we'll post on our website. And for undergraduate students, there are a lot of different internships and opportunities at MIT, a range of different programs, one of the major ones being the MSRP student. And we have 6 undergrad interns in our lab this summer from all around the country. So that's probably the best way to do it.
Go ahead.
My question was talking about your biology for robots. A lot of your work was on the small scale, so I was wondering if you could scale it up, like if it's something that's scalable in like a short-term like future, and if you could scale, like pretend like it was large, like what functions would you want it to ideally do?
Yeah, that's a great question. So one thing I didn't have a chance to talk about is that a really big challenge, actually, not only for the robotics, but also for the healthcare stuff, is the scalability aspect, right? Because usually if you want to replace a large piece of tissue, or you want to make a robot that's like me scale doing something useful, you want to be able to make bigger tissues. The reason we make tissues as small as we do, part of it is like time and cost. Cells are expensive. It takes time to grow millions or billions of cells that go into each of our robots. But the bigger reason is that we don't have any blood vessels in our tissues, right? So if they're 0.5 millimeter thick, which is how thick they are right now, the amount that sugar from the outside liquid can diffuse into cells is about 250 microns, so 0.25 millimeters. Anything thicker than 0.5 millimeter, then all the cells on the inside will be dead. So in order to make things bigger, we need to be able to put a vasculature into the muscles, which is something we are working on right now. And interestingly, one of the questions kind of similar to our putting nerves and muscles together. We're starting to look at how exercise can be used to accelerate the growth of blood vessels into muscles. So if we can make them bigger, then we can make bigger robots that do useful things like gripping and functioning. But we can also make bigger grafts for medical implantation.
So you said like this field is kind of at its beginning, and these like the examples you showed us kind of have to be in a petri dish with certain conditions. Is there any applications of this in like the close future where it can be deployed?
Yeah. So something that I would say a couple things. So on the medical side of things, we're talking to some folks at Boston Children's Hospital about helping babies that have traumatic injuries during birth, thinking about whether we can stick electrodes in their arms, stimulate the muscle, and use that to regrow nerves that have been damaged during birth. So that's sort of an earlier, but would still take us like a decade optimistically. And it should, right? Because whenever you're doing something like this, you should take your time and do it safely and well. Another sort of early stage application that we're looking at is with a lab in London that develops a lot of surgical robots. So if you think about like the next generation surgeries, many of them are not just humans, right? They're humans and robots kind of working together, like teleoperated surgical systems. And those robot surgical things, they have little fingers, but they're all metal. And so they don't manipulate as well as a human can. They're harder, they damage the tissue. And so what we're trying to do is replace those end effectors, they're called like the fingers for the surgical robots, with our muscles to see if surgeons can sort of use their muscles and robot muscles together.
So if you were to stimulate the muscle using the neurons, if it was to do a motion that might normally hurt the person, like if they have, for example, like ulnar nerve damage and they're trying to like move their wrist around, would stimulating it with the like for example like your wristband, would that also hurt, or would that not be recognized in the brain?
Yeah, it's a good question. I mean, it really depends probably on the person's specific disease. So if they have something where they have increased pain sensation because their sensory neurons are like firing too much, then yes, it would still hurt because that would still be happening. If they are lacking motor control because there's some communication gap between their nerves and their muscle, and then you stimulate it externally, then it wouldn't hurt. So it would really depend. And so then usually whenever you make a therapeutic, it's never like a one-step solution. It's like this for this person in this context, and then what other supplement would you do? So maybe if somebody has increased pain sensation, having something that blocks or dampens the impact of sensory nerves in addition to this would be useful.
When you first start growing the muscle tissue, like before it proliferate, did you extract that first muscle cell from some other animal?
Oh yeah, that's a great question. So we use 3 different kinds of muscle in my lab. So all come from different sources. So most of the robot work I showed you is from what we call a mouse cell line. So that means like there's this person named Helen Blau, and she in like the 80s got some cells from a mouse that just grew really well in a petri dish. And she basically made like a cajillion of them and froze them down in vials, and we can keep growing and expanding them. So they were originally from a mouse, but like decades ago. So that's what we call a cell line, where it's basically like a renewable resource. There are some tissues we use in our lab, which are what we call primary muscles. So we take them either from an animal or from a human from scratch. There's reasons for doing that, particularly in disease modeling, if you want to study that person's specific muscle. And then the third kind of muscle we use in my lab is sort of a hybrid between those. It's what we call an IPS cell, so induced pluripotent stem cell. And what that is you have an adult human, you take some of their skin cells, you reprogram them to be in a stem cell-like state. This is a technology that won the Nobel Prize like 20 years ago. And then once they're in the stem cell-like state, you redifferentiate them to form muscle. So it's a very complicated and expensive process, but that's also something where it does originally start from a person or an animal, but then it's in a vial, and that's how we usually get it. It's like frozen down, and then we thaw it, and then we grow them.
Yeah, so hello. So throughout your talk, you talked about how you use light to control the muscles actuators. So I was wondering, why would you prefer to use light? And could you elaborate more on how this works? And also why you would prefer using light rather than electrical signals?
Yeah, great question. So I'll start with how it works. So each cell, right, cell membrane, in our cell membrane, we have many different ion channels. Normally, muscle cells have a calcium ion channel that is voltage gated. So when they get an electrical stimulus, that ion channel opens, calcium flows in, and then a lot of biology happens and the muscle contracts. There's some good explanations of this online if you want to find it. So that's the normal ion channel. And what we use is the same ion channel, but a version that's found in algae. So it's a calcium ion channel that opens and closes in response to blue light. And so that's what we do, and that's how that works. However, the reason of why we want to use it, there's a few different things. So one of the reasons is that electrical stimulation of tissues in culture is not great for them. It causes a lot of bubbling in the media. They get really stressed out. It creates a lot of reactive oxygen species. People are working on making like better, softer, tinier electrodes that kind of mimic nerves a little bit better. But we were having a lot of trouble with it. So we thought with light, you don't have to touch it. You don't have to get in the media. And so it's a really non-invasive way of keeping them happy. And it's worked really well for us. And you can also sort of precisely focus it in different ways without like sticking an electrode in some place. One thing we could do is integrate electrodes with our tissue. So now we're working with some soft electrodes that are working a little bit better. And the other way to do it, which is the sort of bigger and probably best way to do it, is with nerves. And then, because then you have each nerve can control like a single muscle fiber. So you have the most precise stimulation in that way.
Thank you.
Over here. So I was really interested in like how, and I was impressed by like how difficult it actually is to grow these muscles. And I was wondering if growing them like more naturally, almost like already from another animal and using them kind of in their like current form, is that outside the bounds of ethical or practical?
Yeah, that's a great question. So the first paper in this field of biohybrid robotics, which we call it, was from Hugh Herr's lab, who is also at MIT. He's in the media lab. And that paper was published in 2004, where they took a muscle from a frog and basically attached it to a skeleton. And it was really good because the muscle had basically been made in an animal. So it was way better than what we could make ourselves. However, there's some ethical concerns. There's some sustainability concerns because you're making like a whole animal for really a few muscles. But the bigger thing for me, I think the ethical things are very important. But another thing is from the perspective of an engineer, when you're building a robot, you need to be able to know how much force your actuator is going to produce. And we all know that all of us are very different, right? So an animal, you can put it on the same diet, same exercise, there's going to be a ton of stochasticity and variability in how that muscle turns out. So then if you need to redesign your robot for every single muscle that you get from every different animal, it's like not the best way of doing it. So that's the reason why we use this approach instead.
Last question.
Do you have problems or conflicts with the immune system, or you have considered that when you are applying that muscles to like the mouse or the frog?
Could you repeat that? Sorry.
Have you had conflicts or problems with the immune system when you attach the muscle?
Yeah, that's a great question. So there definitely, you want to be very careful about species. So when you're implanting something from a mouse in a mouse, if it's not the same mouse, you will definitely have problems with immune rejection. Even if it's still a mouse cell, but a different cell, you'll still have that issue. So this is something that I would say is not something we're actively working on right now, but there's a lot of different folks in this field that work on thinking about, for example, those iPS cells I mentioned before, is like you could take skin cells from that person, turn them into muscle cells, make a graft, implant them, and then also probably supplement with different types of immune tamping down kind of therapies to sort of make sure that the graft is integrated into the person. So it's a very, very complex problem. So part of it is using the right cells that are specific to that person. And another part is also just learning more about how the body reacts to implants that you put inside for a long period of time.
Thank you.
Thank you. At this time, we're going to transition into our virtual t-shirt presenters. Can we please spotlight Aishanvi and Sahana from Cogworks?
Hello, I'm Shamya Karthikeyan from the Autonomous Cognitive Assistant or Cogworks course. And thank you so much, Dr. Raman, for your presentation. It was really inspiring, and I especially really liked learning about using like engineered muscle grafts to help regenerate like abilities, especially like in the mice legs. That was really interesting, and it was really inspiring for me.
Hi, I'm Sahana. I'm also from the CogWorks course. I really enjoyed learning about the electrical neural stimulation of muscles and how that can be used to mimic exercise and benefit biological systems in real life. We would like to thank you by presenting you with a BWSI t-shirt. Okay, it's blurred.
Whoa. Thank you so much. I like how it appeared off of the screen in real life. Very exciting.
That's all we have for today. So virtual students can log off. Thank you guys so much.
Thank you.
So while we're going to get set up.
In-person students